Hello, everyone, and welcome to today's session. So today's session is about CNS, the cyto and myeloarchitecture of the brain cortex. Now, what it is and so forth, I will explain to you in later slides. So let's dive into it. First of all, you need to know the layers of the brain. The layers of the brain are divided into four main ones and with some sub-main ones. The main one are four: the skull bone or periosteum, the dura mater, arachnoid mater, and finally the pia mater. Now, what is the skull bone? The skull bone is basically the periosteum. You guys already know on it. It's the most external layer, and above it is the scalp, or more external to it. It has two laminas, which are connected with the dipole, and then we will have the dura mater. Now, the dura mater, it's basically a fibrocartilaginous connective tissue, which is coming from the mesenchyme cells, and it has periosteal and meningeal layer. If you guys can look at this picture here, between the skull and the periosteal dura mater is a space. This space is called the epidural space. And then there will be the meningeal dura mater, the periosteal dura mater, meningeal dura mater, and then after meningeal dura mater, there is a space between itself and the arachnoid mater, which is called the subdural mater space, or subdural space, my bad. Now, in the epidural space, so here, there will be mostly spinal cord and a lot of thin-walled vein plus areolar connective tissue. While in the subdural space, there will only be some veins in that area. Then, and the sinuses is also in that area, okay? It's very important to know. Now, let's go to the arachnoid mater. The arachnoid mater, which is this one, it has basically loose connective tissue, some flat cells, and it helps, or in that matter, is where the arachnoid villi, the trabecula from the arachnoid villi, will produce the arachnoid granulation, and arachnoid granulation will basically drains the CSF to the subdural sinuses. So this CSF can go to the circulation, blood circulation, and can get recycled, and a new one is obviously getting produced by the arachnoid mater already. Arachnoid plexus, my bad. And that's it. Then we go to the pia mater. The pia mater is, again, loose connective tissue, but it's very, very vascularized, guys. Quite vascularized, and that's why it's where we can find the blood-brain barrier. Now, because it's a very important area, and it will have a bit more astrocytes compared to other areas for protection of its axons. Now, next, we will have the two divisions that we talked about, the cytoarchitecture and myeloarchitecture. The cytoarchitecture is basically sort of a division in the cerebral cortex based on the shape and the size of cells in that area. And as we already talked about it, because there is a lot of ribosomes in that area and a lot of ER in the plasma reticulum, they're together called the nasal bodies, and nasal bodies can easily be stained with nasal staining. Now, let's go back to the division. It's divided into 6 different layers. The first layer being the molecular layer, then the 2 external, and then 2 internal, and finally multiformis layer. The 2 external, first external will be external granule layer, then external pyramidal layer, and then again, going back to the first name, internal granular layer, internal pyramidal layer. So it's one way for you guys to memorize it. And then multiformis layer. Now, the molecular layer, it basically has, most important cell you need to know about it is the horizontal cells of Kajal. And it is overlying already the pia mater. And we will have the external glandular layer. By the name, you guys can understand there will be some pyramidal cells, small, very small ones, and then there will be the external pyramidal layer. Now, the external pyramidal layer has medium-sized pyramidal cells. I know you guys gonna read quite a lot of other cells in that area, but I don't believe you need to mention them. They're this like small, medium-sized pyramidal cells is enough for this area. And then for the internal granule layer, obviously, again, granule cells are there, small granule cells, and they're connecting the pyramidal cells together. However, you need to remember that there are some pyramidal cells in that area too. Finally, we reach to the internal pyramidal layer, which is the largest pyramidal cells of the body are in that area. They're called the bed cells, and they're dendrites basically are extending to the periphery, and they're communicating with the granular layer here and there. And finally, their axons are extending to the white matter. Now, we will have a multiformous layer. It's another name for so many different cells in the same layer. And the cells could be, the easy names that you can remember is fusiform cells, granule cells, and stellate cells. There are more, but these three will solve your problem for answering this question. Now, these are the cytoarchitecture of the cerebral cortex. Now let's go to a more easier division, which is the myeloarchitecture, which is based on the myelination of the fibers in the CNS of the cerebral cortex. Now, as we already talked about it, the cell responsible for myelination in the CNS is the oligodendrocytes. So, and we already know that myelination is what causes the formation of white matter and gray matter. Whatever is myelinated is in white matter, whatever is not is in gray matter. What structure get myelinated? Obviously, the axons by the oligodendrocytes. So the axons will be on the white matter. That's it. Then the gray matter will be remaining. So the cell body and the dendrites will be on the gray matter. And astrocytes obviously will be there. Microglia or macrophages of the CNS will be in that area, or macrophages of the whole nervous system is called microglia. Anyway, and also cortical regions of the cerebrum and cerebellum are also gray matter. And also you will find the anterior and posterior horns of the spinal cord, which the anterior one is responsible for your motor responses, and the posterior one is responsible for your sensory responses, sensory information to be coming to the CNS. And with that being said, this is the end of our session today. I hope you guys understood it. And as you can see, the question is very long, but the answer is very simple, and you can easily get the point if you just know this simple concept of how to answer this question. And my email is here. If you guys had any questions, I'll be happy to help you. See you guys soon.